Okay, guys. So we're continuing with genetic expression. Today we're going to be talking about how does the cell. Today we're going to be talking about how the cell turns on and off some genes, and as a result, the cell is capable of regulating what proteins are being generated. And these proteins, we said, they are responsible for cellular structure, cellular integrity, and also cellular physiology, cellular function. So we are continuing with the central dogma of biology, and in the central dogma of biology, we begin with a nucleotide polymer known as the DNA. So that's a nucleotide polymer. And from that, we transcribe another nucleotide polymer known as RNA. This is the direction of information flow from the DNA, the nucleotide polymer, into RNA, which is also a nucleotide polymer. And from the RNA, we're going to get translated into the proteins of interest. The protein that is desired, the protein of interest. And we have one of two types of proteins. We either have a structural protein or a functional protein. Structural protein is such as collagen. So that's the first example of structural protein. And where is collagen found? It is found in the ligaments of the bone. And secondly, we have keratin. That's another type of structural protein. And keratin is found in the skin and the nails. An example of functional protein would be myoglobin. What's the difference between myoglobin and hemoglobin? Myoglobin is important for oxygen storage, while hemoglobin is important for oxygen delivery. So if hemoglobin and myoglobin both arrive to the tissue, and both of hemoglobin and myoglobin arrive to the tissue carrying oxygen, only one of them will unload the oxygen and give it to the desperate tissue needing oxygen for aerobic respiration, and the tissue needing oxygen for oxidative phosphorylation. Only hemoglobin is going to give it, and myoglobin will never give the oxygen because it does not transfer oxygen, it only stores oxygen. But in the end, it's a protein, and there is no way, it's impossible to get protein unless when we translate mRNA, and the process is known as translation. And it happens in the cytoplasm. And when we study the cell compartments, we have seen that the cell is divided into three major compartments: the plasma membrane and everything that's in it, and the cytoplasm and all the organelles and the cytosol that's in it, and then the nucleus and everything that's in it. So in the cytoplasm, we have two things. We have the cytosol, which is the fluid part of the cytoplasm compartment. It is white gel that is a colloid because it contains a lot of proteins. And we have all the organelles. One of which is our ribosomes. The ribosomes, they are responsible for translating the RNA. So the ribosomes will translate the RNA, and we have many other organelles. We talked about the peroxisome, that's the shredder of all the misfolded and the improperly folded proteins. We talked about Creutzfeldt disease, as well as mad cow disease. These are the same exact diseases happening in two different organisms. And what happens is you have improperly folded and misfolded proteins that deposit and accumulate at very high levels that they cause stress and damage to the nerve tissue in the brain. So this is an example of what happens when proteins are not properly folded. And here is where the proteasome will come, and it's the shredder of all of these improperly folded proteins. It would destroy them. But in Creutzfeldt disease, or for example, when there is cocaine abuse, what happens is the rate of the accumulation and the deposition of these misfolded proteins is so high that it is overwhelming to these proteasomes, and then there is a transport maximum. Then the ceiling effect has been reached, and then we have a turnover of so many more misfolded proteins damaging the cell. So all of these organelles are laid out in the cytoplasm, which is, as I said, different from the cytosol. So translation happens in the cytoplasm, while transcription, the process of going from DNA, transcription, the process of going from DNA to RNA happens inside the nucleus, So the cell is able to survive. The cell is able to carry out metabolism, the process of catabolism and anabolism by protein enzymes. These protein enzymes, they are responsible for carrying out the process of catabolism and carrying out the process of anabolism. When to turn which protein on, when to turn which protein off, that is genetic expression. So let us talk about the importance of the enzymes. You see, these enzymes are biocatalysts. What's a biocatalyst? A biocatalyst is an enzyme that speeds up biological reactions. That does not mean all the enzymes are speeding up biological reactions. There are enzymatic reactions. There are physical reactions. But the reaction like glycolysis, the breakdown of carbohydrates to give us ATP, and the breakdown of lipids through the pathway of beta oxidation to give us ATP, the catabolism of proteins, and the opposite, the reverse, the buildup, the anabolism of lipids, the anabolism of proteins, and the anabolism of carbohydrates. That requires enzyme catalysts who will speed up the reaction. How do the enzyme catalysts speed up the reaction? So in our reaction coordinate, in this reaction coordinate, we have an x-axis and we have a y-axis. On our y-axis, we have the delta G, the Gibbs free energy. So this is the free energy. Okay, and x is what happens as unit time. I still see some students having problems with the difference between exergonic and endergonic. So exergonic and endergonic is describing to me the state of energy within the bond. So if I am building up, if I'm going from amino acid, micromolecule, amino acid is a micromolecule. It is a monomer. If I go up to a protein, and this protein is a macromolecule, a polymer. When I go up, I am saving energy. I'm storing the energy. So if I ate pancake, if I had a meal, then I'm going to break down that food that I just ate into its building blocks. Okay, if I ate a sugar, I'm gonna break it down from from polysaccharide, break it down from polysaccharide into disaccharide, and then finally into monosaccharides. So I built it up into its monomer form. And if I eat a protein, I break it down from a polypeptide. I break the peptide bond down into an amino acid micromolecule. If I ate a fat, if I ate a lipid, if I ate steak, I break that down from a lipid all the way down into free fatty acid and glycerol. So when I break everything down, I get energy. So what happens to the bond? When I break the bond, the energy exits the bond. So that's exergonic. Where did it exit the bond to? Exergonic. It exited the bond to me. So now I'm running the marathon. All the sugar that was stored in my liver, all the steak, fat, lipids that were stored in my adipose tissue in my belly, all of the proteins that were stored in the liver are going to get broken down. The energy, the peptide bond, the glycosidic bonds, all of these bonds are going to get broken. When they get broken, the energy is going to exit the bond. That's why we call it exergonic. And the bond has no more energy. That's why it's negative delta G, because it no longer has energy. So in catabolism, in breakdown, when I'm breaking down polymers into monomers, I am removing energy from the bond and I'm utilizing it for myself so that I can run the marathon, so I can work and burn energy. But the opposite, if I no longer now, I had a meal, but I'm not burning it, I do not need energy, what's gonna happen is I'm gonna need to store to make the bond. So if I make the bond, the energy is going to exit me and it's going to go into the bond. So now we're going to form the bond, and that's known as plus delta G, known as endergonic. So this is a convention. Endergonic, exergonic is a convention to describe the state of energy in the bond, in the macromolecule. Am I breaking down macromolecules? Then there is no more energy because I broke down the macromolecule. The energy comes to me. That is known as delta G minus delta G, exergonic. But if I form the bonds, then I do not need energy. I'm storing the food in the form of energy in these bonds, and that is known as positive delta G, endergonic. Clear? Okay, so that's very important to know the difference between exergonic and endergonic. Anybody has a question? Does anybody have a question on the difference between endergonic and exergonic? Crystal clear. Okay, so if we go back into our reaction coordinate, this reaction coordinate is depicting for me energy as a function of time. So if I have a reaction where I have reactants here, and the reactants need to be converted in products. Well, we know that that path is not smooth. We have to go uphill. We have to go uphill. We have to go uphill. When we have to go uphill, there's two kinds of energy now we have to recognize. The energy between reactants and products, that is known as the net free energy. The energy between reactants and products, that is known as the net free energy. But the energy between reactants and the transition state, that is known as the activation energy, EA. So what the enzyme will do is the enzyme will provide us with an alternative pathway where we go from reactants, into products, and we lower the activation energy. So the activation energy with the enzyme, EAZ, the activation energy with the enzyme gets lowered down, and that's what's going to make the reaction go really fast. But if you look, is the net free energy affected at all? Is the net free energy affected? It is not touched. We only lower the EA. We don't lower the NA, the net energy. We don't lower NE. We don't touch the net free energy. It's unaffected. What we change is the activation energy. We no longer have to go up through a mountain. We just gotta go up through this hill. So the top point here, this is known as the transition state. The top point here, this is known as the transition. This is known as transition state. This is known as transition state. And the top point up here is the saddle. This is the saddle point, the highest, the top point. So we're gonna lower that saddle point to a new alternative saddle point, and that's what's gonna make the reaction go faster. So you must remember that the enzyme, number one, does not affect the net free energy in purple. Number two, it does not get consumed in the reaction. So the enzyme is always going to be there, ready to be used more and more and more, and to catalyze more reactions. As long as you pay it in ATP, if there is no ATP, enzymes will only work if there is energy in the form of ATP. If you provide energy in the form of kinetic energy, it will not work. The enzyme will not work with kinetic energy. The enzyme will not work with potential energy. The enzyme will not work with electrical energy. The enzyme will only work with chemical energy. So, and it does not get consumed. And number three, it does not alter. It does not alter the reaction. It does not alter the reaction. So let's say I'm supposed to have this type of reaction. A and B plus C. A and B plus C. And I'm supposed to get A and C plus B. The enzyme is not going to say, well, let me give you something else. Let me give you AC plus D. It's not going to give you something different. It will give you the same product, just faster. All the enzyme will do, it does not alter the reaction, does not change the product, okay, does not get consumed in the reaction, does not affect the net free energy. It only speeds up the reaction by decreasing the activation energy. So on the exam, students always, when they get a question like the enzyme is a biocatalyst that speeds up the reaction by decreasing the net free energy, true or false? They say true. But it's not the net free energy. The net free energy stays the same. It is the activation energy between the saddle point in the transition state complex and your reactants. So this transition state is a complex where you have weak bond between all of your reactants and products. meaning that you are not a reactant, but you are not yet a product. So you are in transitioning. You are some way in the middle. Thank you. So these enzymes are proteins. And we said the law says you're never going to get proteins unless you have RNA. And you're never going to get RNA unless you have transcription. So when we move inside the nucleus, then we find out how the cell is able to transcribe proteins. Thank you. That's how the cell is able to transcribe mRNA to get the proteins of interest. So you have to understand that these enzymes, they are the ones who catalyze and initiate these important reactions. But these enzymes are off. They have to be turned on. So if somebody had a meal and somebody is hungry, the enzymes who are on in their bodies are different. One has enzymes that accelerate the hunger sensation and the search for food. The other one has hormones that are different, that are initiating satiety and feeling full. But these enzymes have to be turned on. So how are these enzymes turned on? One of the ways by which enzymes get turned on is through an enzyme system known as the protein kinase. What's a protein kinase? Protein kinase is an enzyme that adds a plus pi. What's a plus pi? A plus pi is a phosphate group. So you can have an enzyme that is off. This enzyme will be turned on if you add a plus pi. If you add a phosphate group to it, it's going to get turned on. So who added the phosphate group? It's this kinase. So once you activate the kinase, the enzyme is going to get a plus pi and then it's going to start to work. Are we clear?
Wait, so the protein kinase adds the phosphate?
Yes, it's an enzyme that catalyzes the addition of a phosphate group to the enzyme. Some other enzyme is the other way around for them. Some enzymes are on, but when the kinase adds a phosphate, a plus pi to them, a phosphate group to them, they're going to get turned off. But all you need to know for now is that what is a kinase? A kinase is an enzyme that once activated will add a phosphate group to another enzyme. Adding that phosphate group to another enzyme may get that enzyme to start working or will get that enzyme to be inactivated. These enzymes, they can be inhibited. We can inhibit them, stop them from working. These enzymes can get activated. We can activate them to start working. So how do we sustain life? How do we propagate these biochemical essential life reactions? Through the enzymes. So if you are a vegetarian, you add a veggie meal, somebody else had steak, someone else hungry. The enzymes in the body of each one who are activated are different. So it is those enzymes who will give us the essential metabolic activities that are happening in our bodies. So if you remember when we talked about what happens, this is very important. When we talked about what happened in the phospholipid bilayer, we said that the cell membrane is made up of a phospholipid bilayer. The phospholipid bilayer is actually made up of, it's a triglycerol, three carbons, each carbon has an OH, each carbon has an OH, each carbon has an OH, and a free fatty acid, free fatty acid with an OH and a bunch of carbons, free fatty acid with an OH and bunch of carbons, free fatty acid with an OH and bunch of carbons. And what happens is we do what to join them together? If we want to join them, are these micro or macromolecules? In that shape, are they micro or macromolecules? They are micro. So that's free fatty acid 1, free fatty acid 2, free fatty acid 3. And this here is what? What is this complex here? Glycerol. So that's glycerol. So this glycerol is a separate molecule, glycerol, separate molecule from these free fatty acids. If I want to join them, I remove an OH and an H from here, and then water will come out. So I remove them, and then they get joined. They get joined, 3 carbons, okay, and water came out, so they will be connected like that. So now they are connected. So now that is no longer a micromolecule. That's a macromolecule. What do we call that? After they assemble together, what do we call that? A triglyceride. That's a triglyceride. Okay, this triglyceride, how do I modify it and put it in my phospholipid bilayer? I remove one tail, and instead of the tail, what do I add? I remove one tail from the triacid glycerol, and what do I add? Excellent. I add a phosphate. With a minus charge. Then I add a group of choline, and that is my phospholipid head, and these are my 2 tails. These are my 2 tails, and that's my red head. This is the red head. This is the red head. That's my phosphate and choline. This is the phosphate and choline. And the 2 lower brown tails, these represent these 2 lower tails represent these are non-polar. But this is polar head. This one side of my glycerol is a polar head, and these other 2 are my tails. So these are my 2 tails. 2 tails, 1 head, 2 tails, 1 head, 2 tails, 1 head. These are my 2 tails, 1 head. 2 tails, 1 head, 2 tails, 1 head, 2 tails, 1 head. Okay, you guys look like you're still confused. That's a glycerol. See, you cannot call that a sugar. That's a glycerol with 3 carbons and an OH and an OH and an OH. That's a glycerol. Glycerol. On the other side, you will have your free fatty acid. You will have 3 free fatty acids. These free fatty acids are carbonyl with an OH, carbonyl with an OH, carbonyl with an OH, and they're all connected to a long carbon chain, a long carbon chain, a long carbon chain, a long carbon chain. They are now, these are 4 different molecules. These are 4 different molecules. Now I need to assemble them. I need to assemble them. How do I assemble them? By minus H2O, by hydrolysis, minus H2O. So I remove an OH from here and an H from here, and I connect the bond like that. I remove an OH from here and an H from here, and I connect the bond like that. I remove an OH from here and an H from here, that's hydrolysis, and I connect the bond like this. Now I no longer have 4 different molecules, I have one big molecule, and that is known as a triglyceride. A triglyceride. That is a triglyceride. So how do we modify that? And we can plug it in the membrane. How do we modify that? We take one of the tails out. We keep 2 tails, but we take one of the tails out, and we add a phosphate group that is negatively charged. We add a phosphate group that is negatively charged. We add a phosphate group that is negatively charged with a minus charge, and a choline, choline, and a choline. And that is my head. And these 2 are my orange tails. This is my orange tail. 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 And this is my phospholipid head. This is my phospholipid head. That's my phospholipid head. That's my phospholipid head. And these are my 2 tails. That's what I have here. That is what I have here.
Can I ask where the choline came from?
The choline is just assembled. It has a nitrogen. So there's an enzyme that catalyzes just that. The process of cutting one of the tails and replacing phosphate and putting choline. The choline stabilizes the phosphate group, and both of them are polar. But they make my head, my red head. This red head here is polar head because it has the phosphate and the choline. The nitrogen with an N. You see how I drew the N. The nitrogen and the choline. So that's my plasma membrane. That's my plasma membrane. So now you see how we got 2 tails, 1 head? That's the red head. That's the red head. That's the red head. And these are the 2 orange tails. Okay, are we clear with that? In the back, are we clear? These are my 2 tails, orange tails that are coming down, the 2 tails, and this is my red head. Okay? Now, let's see what happens with okay, then we have on the other lower leaflet, we have also red heads. This phosphate choline, phosphate choline, phosphate choline, phosphate choline, phosphate choline, phosphate choline, phosphate choline. That's the head. Phosphate and choline, 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 phosphate and choline. So that's my 1 head, and these are my 2 hydrophobic tails. One is polar head because it's minus phosphate with a choline, and I have my two tails who are my 2 tails who are non-polar. My 2 tails who are non-polar. Okay, this is very important. We said we have classes, we have very important classes of hormones that are supposed to transmit and mediate for us. They're supposed to transmit and mediate for us an intercellular target effect. I need a target effect. So, how do I get a target effect? I get it through my hormones. My hormones. How many different types of hormones do I get? I have the first type, these are steroid hormones. And these are cholesterol based hormones made of cholesterol. Cholesterol itself is not a hormone, but the hormones are based on cholesterol. Like dough, you make from dough pancake, you make from dough cake, you make from dough bread. It's the main raw material. But the steroid hormones are your aldosterone, your testosterone, your estrogen, your progesterone, and so on and so forth. These hormones are first messengers. These first messengers, these hormones, they can dive. They are welcome to go through the bilateral leaflet. This is nonpolar. This is lipid. This is lipophilic. This is polar, this is nonpolar. And so is cholesterol. This is also nonpolar. This is orange also. I should have done this in orange. This is nonpolar. This is lipophilic. So what happens now, if the steroid hormone is targeting my cell for a target effect, it is welcome. It is welcome to come through because it can dive. It can dissolve in this bilateral leaflet. It will come through and then it can swim in my cytosol and it can enter my nucleus. And in my nucleus, I'm going to have my DNA. And on the DNA, there are receptors that are bound to the chromosome, to the DNA. And once this aldosterone, testosterone bind to the DNA, it's going to be able to transcribe for me mRNA. Then mRNA is going to get out and get translated. Translation in the ribosome. And once it gets translated in the ribosome, it's going to give me my protein, my target protein, and my target protein is going to give me my target effect. So what are we saying here? We're saying here that when you are a steroid hormone, you do your job by yourself. You don't need a second messenger. The first messenger is able to cross the bilateral leaflet and cross the nuclear membrane and bind to the chromosome and is able to transcribe mRNA, and the mRNA that has been transcribed is going to generate for us a transcript that will be translated, and then will give us the target protein, and the target protein will give us the target effect. So because we are nonpolar like the bilateral leaflet, we did the job by ourselves. Now look at what happens with epinephrine. Epinephrine is not steroid based. This is known as catecholamine. Catecholamine hormones like epinephrine. Why is epinephrine important? Epinephrine is for fight or flight. Epinephrine is the hormone of stress, like adrenaline. That enzyme provides you with energy during stressful conditions so you can survive a form of stress or a form of anxious bout. So what happens is epinephrine is a catecholamine. It is polar. Okay, it is lipophobic. So can it enter? Can it cross? It cannot cross. So what it will do is it will bind a peripheral protein out here. It will bind a peripheral protein out here. It will bind a peripheral protein. Then there is an attached transmembrane receptor that is an integral protein. Attached integral protein, transmembrane receptor, integral protein, transmembrane receptor, and the transmembrane receptor is going to start making some conformational change. It will make conformational change, and this conformational change is going to cause the activation of an internal peripheral membrane protein known as G protein, and the G protein is a peripheral protein, and this G protein is going to start migrating until it finds a membrane-bound enzyme known as adenylate cyclase, okay, and it's going to cause adenylate cyclase to switch ATP into, remember it's called adenylate cyclase. It's going to convert adenine triphosphate into cyclicized adenine monophosphate, cyclic AMP. This cyclic AMP is my second messenger. That's my second messenger. The second messenger, the cyclic AMP, is going to go and it's going to activate for me a protein kinase. And the protein kinase is going to go and phosphorylate, is going to add a plus PI, is going to add a phosphate group to an enzyme, turning the enzyme from off into on, activating the enzyme, turning the enzyme from off into on, and then that enzyme is going to give me my target effect. That is my target enzyme. That is my target enzyme, and it gave me my target effect. So the difference is, if you are nonpolar, if you are lipophilic, you are just like your orange, just like the bilateral leaflet, you're so welcome to cross through. So as a first messenger, you don't need a second messenger, you don't need no cyclic AMP, you can go yourself, you can bind inside the nucleus, transcribe your mRNA, mRNA is going to get transcribed, translated into protein, the protein is our target protein, is going to give us our target effect. Anabolism, catabolism, bring energy down, bring energy up, and that's what it's going to do, that target protein. But if you are a catecholamine, like epinephrine, and you are polar, you have to bind on the outside. You're not welcome to come inside because you're not orange. You're not lipophilic, you're lipophobic. You're not nonpolar, you are polar. So you cannot cross. You will stick to the outside. And once you stick to the outside, you're unable to come through, and you will be staying on the outside, binding a receptor protein. When you bind the receptor protein, you're going to dimerize. Then an internal peripheral protein known as G protein is going to go and going to activate adenylate cyclase. Adenylate cyclase will convert ATP to cyclic AMP, and then cyclic AMP is going to activate a protein kinase, and the protein kinase is going to add a phosphate group to my target enzyme, and my target enzyme will give me the target effect. So here I needed a second messenger. I couldn't do it myself. I needed a second messenger. I could not have done the target effect like here. I needed a second messenger. So the orange steroid hormones, they can go straight to the nucleus, or they can bind to a receptor in the cytoplasm, and then that receptor is going to mobilize. This is called the mobile receptor. The mobile receptor hypothesis. The mobile receptor hypothesis is going to cause the mobile receptor to move from the cytoplasm into the inside of the cell and carry the central dogma of biology. Go from DNA, transcribe RNA, from RNA, translate to my target protein, who will give me my target effect. So you have to know your polarity. So that's very important. So now we have seen that the cell is responsible for turning on and off enzymes. Now let's see how the cell is also responsible for turning on and off genetic expression. This process here. So from person to person, transcription is not the same. So they did an experiment. This experiment is very important for you to understand. In this experiment, they took two cells, an ovum cell from the frog. Ovum cell, that's the egg cell, the female cell. And that ovum obviously unites with the sperm, and then fertilization will happen. And when fertilization happens, then cell division will take place, and then there will be development. So they had a skeletal muscle cell from the frog, and they had a ovum cell from the frog. So obviously the cell must have a nucleus. And this is the skeletal muscle, must have a nucleus. So what they did was they removed the nucleus of the ovum cell. So now the ovum cell has no nucleus. And they took out the skeletal muscle cell nucleus and they planted it inside the ovum. So now the ovum does not have an ovum nucleus. It has a skeletal muscle nucleus. What do you think happened when the frog sperm came? Frog sperm. What did it do? It fertilized the egg fine, as if that nucleus was actually a skeletal muscle nucleus. So what did they conclude? They concluded that it is, for example, if you have $1,000 and you give the $1,000 to a chef, the chef is going to go buy cooking utensils. If you have $1,000, you give it to an athlete. The athlete is going to go buy sneakers and tracksuit, and is going to go buy that athletic stuff. If you give $1,000 to a police officer is going to go buy, for example, gun, going to go buy weapons and whatever. So that shows you it is not the $1,000. It is who are you that is going, how are you going to use that $1,000? So that was a skeletal muscle nucleus. It was planted inside an ovum. Yet everything went fine for the ovum because inside the nucleus you have the DNA. Inside the nucleus you have the DNA. It is the machinery, the RNA polymerase. It is the RNA polymerase will target a certain part that will give the cell its identity. So for example, in the skeletal muscle cell, there is an RNA polymerase of the skeletal muscle cell that will only transcribe the regions, that will give me skeletal muscle cell proteins. It will only transcribe the region that will give me skeletal muscle protein. In the ovum, you have an RNA polymerase that will only transcribe a region in the nucleus, that will give me the machinery, that will give me the machinery for the ovum. Is that clear? Are you guys paying attention? I'm not going to publish I'm going to delete the study guide. I'm not going to publish study guide no more. You guys are not paying attention. I'm talking. I'm trying to record this and post it for you, but people are not participating. People are not even giving eye contact. You're working on the study guide, and we're like, really no learning is happening. I don't understand what's the problem. What is the problem? What am I doing something wrong? So why are people not participating, and why are people not raising their hands? What's happening? Okay, I'm going to go through this experiment again. This is very important. They removed a nucleus from a frog skeletal muscle, and they implanted it in an ovum. And they thought things are not going to work because, that's a nucleus from the skeletal muscle. If I place it in an ovum, things are not going to go fine. But they realized when they fertilized that ovum, everything went perfectly. Fertilization was successful. And there was growth and development. So then we started to have an embryo. So that embryo started to divide. So we had a blastula, and then we had a morula. That's the next stage. And then we had gastrulation. And then you started to have an organism. But you did not have your original ovum. So they concluded that it is not actually the problem of the nucleus. It's the nucleus has the same DNA. So whether you are a nucleus in the skeletal muscle cell, whether you are a nucleus inside the skeletal muscle cell, or inside the neuron, or inside the heart cell, or inside the liver cell, you're still a nucleus and you have the same DNA. Let's say now this big circle is a nucleus. This big circle is a nucleus. This big circle is a nucleus, and it has this is the DNA. They realized that DNA is this section. If you transcribe this section, you will get skeletal muscle proteins. If you transcribe this section, you will get ovum proteins. If you transcribe this section, you will get pancreatic proteins. So it's not the nucleus that gives you your structure and your function. It's the set of the polymerases that is found on all these different cells. So even though the ovum got a new nucleus, it still has the same polymerase. It still has the same team of polymerases, and that same team is going to go inside the nucleus and is going to go to the ovum part. In the skeletal muscle, that team in the skeletal muscle, if the nucleus was inside a skeletal muscle cell, the polymerase is going to transcribe the skeletal muscle cell region here. If it's the ovum, it's going to transcribe the ovum region. If it's the pancreas, it's going to transcribe the pancreas region. So the set is the same. That's why I told you, if you give $1,000 to an athlete, you give $1,000 to a police officer, you give $1,000 to a chef, it's the same $1,000. But they all will use it differently because they have a different function and they have a different structure. Same thing with the cells. They have the same nucleus, they have the same $1,000, but they're not going to use it in the same way. This whole DNA is not all of it is active. Only the region that pertains to the cell. So the skeletal muscle cell will transcribe a region. The ovum will transcribe a region. Everything else is heterochromatin. That part is euchromatin. If you transcribe the liver region, it will give you the proteins that are necessary for the liver cells. if you transcribe the bone region, it will give you the proteins necessary for the bone cells. So the DNA is really long, but it's for everybody. You only transcribe, you only go transcribing based on what kind of cell you are. And then you translate what you transcribe into your target protein. Does that make sense? Everybody understands? You understand the significance of devoiding a nucleus from a frog skeletal muscle cell and putting it in an ovum cell and then fertilizing that cell. Is everything going to go fine, or there's going to be a breakdown? Everything's going to be fine. And why is that? Why is that?
Because all the DNA is in the nucleoside. It's just going to get what part it needs.
Excellent. Thank you. Okay, so now we're going to move onward to the different set of enzymes that give us our different polymers. So now we have already talked about how this DNA is inside the nucleus in form of sister chromatids. It's either 46 sister chromatids, or 23 pairs, or 23 pairs, or 23 pairs. Either 23 X's joined together or 46 single. And it has many strands of DNA. So if I'm going from DNA to RNA, what am I going to need? I'm going to need an enzyme, an enzyme. This enzyme is known as polymerase. It's a polymerase. So what am I polymerizing? If I'm polymerizing RNA, I can either polymerize long chains of nucleotides that gives me RNA. If I make a long polymer of RNA, that makes me an RNA polymerase. And if I make a long polymer of DNA, I'm going to need a DNA polymerase. I'm going to need a DNA polymerase to make a DNA polymer. I'm going to need an RNA polymerase to make an RNA polymer. And I'm going to need a DNA polymerase to make a DNA polymer. So if a DNA polymer is made, who made it? It's a DNA polymerase. That's why they call it a DNA polymerase, because it makes polymer of a DNA. Why they call that one RNA polymerase? Because it makes polymer of RNA. The issue here is, what am I dependent on? So in this case right here, if I need to make if the ovum needs to make the proteins that will help the ovum divide and carry out the fertilization successfully, I'm going to go to the DNA region and I'm going to go transcribe that region inside the nucleus. So I'm going to depend on the DNA. So that's called DNA dependent RNA polymerase. DNA dependent RNA polymerase will give me an RNA polymer, this one. So I got this RNA polymer right here. RNA is a nucleotide polymer. RNA is a nucleotide polymer that depends on the DNA. So it is a DNA dependent. it depended on the DNA. This is the DNA strand. And the polymerase went in and transcribed for every G. It put, like let's say you have a G here, C, you have a T, you have an A. So for every G, put a C. For every G, for every G, you put a C. For every C, you put a G. For every T, put an A. For every A, you put a U. And that is your DNA, that is your RNA transcript. So you depended on the DNA. That's why they call it DNA dependent RNA polymerase. But in reverse transcriptase, the HIV virus is a retrovirus that has an enzyme known as reverse transcriptase. And reverse transcriptase is going to transcribe for me. It's going to transcribe for me DNA from RNA. So the virus has viral RNA, and it's going to use the viral RNA and the reverse transcriptase enzyme that it has to generate viral DNA, then use the human ribosome to translate that viral DNA. I'm going to use a reverse transcriptase to retroactively reverse the transcription of RNA to give me viral DNA. So what am I going to use? An RNA polymer or a DNA polymer? Am I going to use an RNA polymerase or a DNA polymerase?
To bring it back to DNA, yeah.
I'm going in this direction. So remember I told you, if I'm going to make RNA, I'm going to need an RNA polymerase. If I'm going to make DNA, think no further. I'm going to need a DNA polymerase. That's why they call it DNA polymerase, because it polymerizes DNA. RNA polymerase, because it polymerizes RNA. So here, I'm going from viral RNA reverse, so I can make viral DNA. What am I going to use? DNA polymerase. What did I depend on? Did I depend on RNA to make the DNA, or did I depend on the DNA to make the DNA? What did I depend on?
RNA to make the DNA.
Yeah, this is my RNA. The virus only has RNA. And it made DNA from the RNA. So it doubled, it transcribed, that's why they call it the reverse transcriptase. It reversed from viral mRNA to viral DNA. So we need a polymer, a DNA polymerase. So it's a DNA polymerase, but it was not dependent on a DNA. It was dependent on an RNA. So it's a RNA dependent DNA polymerase. When we're going in that direction, we need an RNA polymerase that is DNA dependent. And in replication, when we are doubling the DNA, let's say this is my cell that needs to divide. Why does the cell need to divide? So the cell division cycle. The cell division cycle will happen, number one, for growth and development. For growth and development. In growth and development, going from an infant to a toddler, to an adolescent, to an adult, getting bigger, that requires a lot of growth, a lot of cell division. Why else do we need cell division? Why do we need to start the cell cycle besides for growth and development? For healing, and for regeneration of tissue, for healing and for regeneration of tissue. So if a tissue is injured, if I'm injured due to a laceration or a bruise, that tissue is damaged. So your immune system is going to send white blood cells that are going to carve out, perform debridement, remove all that dead tissue, and then when all the dead tissue has been removed and we're going to remove all the infectious microbes, and now that environment is healthy and we need to regenerate new tissue, then we will start the cell division cycle to heal and replace that tissue with new tissue. Okay? The same thing in growth and development. So we need to start the cell division. But how does cell division happen? It happens, cells, as the cell theory says, cells cannot come except from other cells. I am here because of my father was there. My father's there because my grandfather. My grandchild is going to be here because of my child. So cells can only come from other cells. They don't come from nowhere. So this, in order for me to divide, whether for healing and regeneration or whether for growth and development, I am going to need to double my DNA. So that's why in the cell cycle, we double the DNA multiplied by 2. So now we have 2x. How does this happen? This happens with replication. So the DNA is going to get replicated, although the DNA used to look like that. We're going to separate the DNA into 2 separate strands. And we're going to use a DNA polymerase who is going to generate for us another daughter strand. And now I have doubled my DNA. And after I double my DNA, I'm going to start my cell division. So now I've separated into 2 cells. After I doubled my DNA, this is known as replication. So in replication, what happened? This green stuff, what is that? What is the green? Is that a DNA polymer or a RNA polymer?
That's DNA polymer.
I'm gonna divide the cell. That's the division of the cell. So the cell is dividing, so it needs to double its DNA so you can have 2 cells. So that green stuff, that's DNA, that's not transcription. I wrote it here. Replication. I'm doubling my DNA. I'm not transcribing. So when I double my DNA, what happens? I get double. So that is, we use DNA polymerase. We use DNA polymerase for the green. Who did I depend on to make a DNA polymerase? Who did I depend on? The purple. What's that? That was DNA or RNA?
DNA.
That's DNA. So that's DNA dependent DNA polymerase. So you have to know the three. The DNA dependent RNA polymerase when we're going in this direction. And for the virus, it has an RNA dependent DNA polymerase. And for replication, to divide the cell, we're gonna need a DNA dependent DNA polymerase. So you have to break it down into two steps. What polymer am I making? Am I making an RNA polymer? That's the RNA polymerase. Am I making a DNA polymer? That's the DNA polymerase. Then go back and look. Who did I depend on? Did I depend on RNA? Then it will be, if I did, that will be RNA dependent DNA polymerase or RNA dependent or RNA dependent DNA polymerase or DNA dependent RNA polymerase or DNA dependent DNA polymerase. That's for my replication. So the cell, if the cell wants a target protein, it will take this route. DNA dependent RNA polymerase. It will go inside the nucleus. It will transcribe for me a region. Will transcribe for me a region that after I transcribe it, I'm going to get an RNA nucleotide polymer. Then when I translate it, I'm going to get my target protein. I'm going to get my target. Yes?
So this DNA dependent DNA polymerase, is that the what is this? What do you
That's for doubling the cell DNA so I can divide in mitosis.
okay.
So, for example, let's say now, remember we said for cell division, if the cell wants to divide, for cell division, the cell wants to divide for reasons like healing, reasons such as healing, reasons such as regeneration, healing such as healing such as regeneration, healing for growth and development of a child who's growing up, I'm going to need to double my DNA.
Right.
So in my cell cycle, I have two phases. The first phase is known as interphase. 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 An interphase is divided into three stages. Three stages. Into three stages. Is divided into three stages. The first stage is the G1 phase. The second stage is the S phase. The S phase. That's the G1 phase. And that is the G2 phase. G1 phase, S phase, and G2 phase. And after that, I'm going to have my second phase of the cell cycle. So this is all known as interphase. This is all, these three steps are known as your interphase. These are interphase. Then after that, I'm going to have my mitosis. This is known as mitosis. Mitosis. And mitosis is divided into prophase prometaphase, metaphase, anaphase, telophase, and cytokinesis. So in order for me to divide and have two cells, I must go down this direction. I must go clockwise. I must go clockwise. So I start with one cell, with one X here. That's one X. In the G1 phase, that one X is going to go through a preparatory phase to prepare all the proteins and all the enzymes that are necessary, all the proteins, all the enzymes that are necessary for the cell to divide. Once I reach the S phase, I'm going to double the DNA, 2x. That's where the replication is happening. I'm going to replication. I'm going to double the DNA 2 times replication. I'm going to double the DNA. I'm going to double the DNA. Then after I double this DNA, I'm going to go through the G2 phase to prepare for cell division. This all the interphase is preparation for division. Prepare for division. So here now I have 2x. Before I get into mitosis, I'm exactly one cell with 2x. So before I enter interphase, I was 1x. After I went through the G1 phase, the S phase, the G2 phase, I'm going to have 2x. Then when I go through mitosis, I'm going to have my chromosome in prophase. I'm going to generate 2 centromeres who are polarizing to one pole. That's in prophase. In prometaphase, they're going to start to polarize to opposite direction. And these are my chromosomes. These are my chromosomes. These are my chromosomes. In prometaphase, they're going to polarize and start to send out, away. The arrow is going to point away from the centrosomes, from the centrioles. It's going to point away. In metaphase, you will have all of your chromosomes lined up in the middle. That's why they call it metaphase plate. Metaphase plate. Metaphase plate. And these centrioles who are in opposite poles are going to send out in this direction, send out their microtubules in this direction. And the other centromere is going to send a microtubule. That is known as the metaphase plate. In anaphase, they're going to start pulling in the opposite direction. So these are your centrioles, and these are your sister chromatids that have been pulled away. By your asters. So now the pulling is in the other direction. So initially, the microtubules pointed away from the centrosome to the middle, from the centrosome to the middle, from the centrosome to the middle. But in anaphase, they pull the other way so they can pluck them off and remove them from each other. So in anaphase, they have completely separated. Then in telophase, they're going to start dividing the cytoplasm. And in cytokinesis, they're going to have 2 separate cells, each one with its own set of chromosome. And now you end up with two X's, one X here and one X here. So replication is happening here. I'm doubling my DNA so I can prepare for mitosis and divide my two cells. Okay, clear? Questions? Do you have a question? Any questions?
Prometaphase isn't in every single mitosis.
So the mitosis steps are the same. So these are always there. Prophase, prometaphase,
Okay.
metaphase, anaphase, telophase, cytokinesis. But these are the steps where I rearrange my cells in a way that help the cell to divide. But that cannot happen unless I double my DNA. And I double my DNA using a DNA polymerase, because DNA is a polymer. Make a DNA polymerase. And that DNA polymerase, what do I depend on? DNA. So I use a DNA-dependent DNA polymerase to double my DNA. Then I go through my cell cycle. So now I have two cells. I need that for healing. I need that for healing. I need that for regeneration. I need that for growth. I need that for development. That's very important. So that was replication, and that was cell division. Now, so you see, this is replication. I have doubled. So that's happening in the S phase here. That's happening in the S phase. That's happening in the S phase. And in the S phase, you have the separation. So the DNA is double-stranded. DNA is double-stranded. You will bring a very important enzyme known as helicase. Helicase. What helicase will do?
Can I ask a question really quick?
Yes.
I'm sorry.
No problem.
So when you have the DNA and then the RNA, right?
Yes.
DNA dependent RNA polymerase,
Mhm.
right? Is that just for protein making then? What does
The
that have to-
DNA dependent DNA polymerase is to
For this.
double my
Right,
DNA.
but what is the DNA RNA polymerase for?
So that's we gotta come to that. The
Oh.
DNA dependent RNA polymerase is for me to make my proteins, all my
Right,
enzymes.
right.
Yeah.
Okay, alright.
Yeah.
And the reversing way is the same thing to make protein.
The reverse what?
The reverse way. when
The
you
reverse transcriptase, no. The reverse transcriptase,
That makes sense.
in order for me to make proteins, I need DNA.
Okay.
But the virus doesn't have DNA. That's his
Right.
problem.
Okay.
But he has two things that will get him to the DNA. The first thing is RNA. He has only RNA.
Okay.
And the second thing is that reverse transcriptase. So once he get in your cell, he will destroy your nucleus. So now you have no human DNA. The human DNA is not there. And he will keep the ribosome, because the ribosome does not discriminate. As long as it's RNA, she will translate it. Whether it's human RNA, whether it's viral RNA, she will translate it. And he knows that. So the virus is gonna go in the cell, destroy the nucleus, destroy the nucleus of the human, and then he will use his own reverse transcriptase in order to make DNA from his own mRNA. And then when we have viral DNA, he will use human polymerase to make viral mRNA, and then he will take that viral mRNA to the ribosome, and then he will make the ribosome translate that viral protein.
Okay.
So you see how wicked the virus is.
Yeah.
Yeah.
I understand that part.
Okay, so that's very important. Okay, now, where are we? We are here. Helicase. Helicase. What does helicase do? That's the easiest one. Helicase is going to unwind the DNA. Helicase is going to unwind the DNA. When we unwind the DNA, we have two strands. I forgot to tell you something here about There's so much. 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 Okay, but anyways, what can we do? Anyways, that's the challenge. We have to get through it. Okay. the replication, before we go into translation, helicase is for transcription. Helicase is for transcription. Helicase is for transcription.
Wait, are you going back?
We're going back to replication now.
Okay.
There's something really quick that I just forgot. Just a little bit, not a lot. But don't worry, December is gonna get here sometime. Just be patient. Yeah. Okay. All right. So, if I want to replicate my DNA, I have to unwind it. In order to replicate my DNA, this is my DNA. It's double strand. We went through all of that. It's double stranded. So, this replication, in order for me to double my DNA, I need to unwind it. So I use a helicase to separate both strands. So both strands in blue are going to get separated. Both strands are going to get separated. And after they separate, I'm going to have, these are my two original strands. These are my two original strands. The two original strands, obviously the DNA is anti-parallel. Anti-parallel. The DNA is, what does anti-parallel mean? It's like two snakes. One head up, 5 prime, one tail down, 3 prime. The other one is, at the head will be the 3 prime, at the bottom is 5 prime, and then they are connected like that. So when I unwind them, one is going to be 5 prime to 3 prime, and the other one is going to be 3 prime to 5, 3 prime to 5 prime. So now this is my DNA. Replication will only happen from the 5 prime direction to the 3 prime. So your DNA dependent DNA polymerase is going to be working and making the daughter strand in this direction like that. For every A it will have a T, for every A it will have a T, for every G it will have a C, for every C it will have a G, for every T it will have an A, for every A it will have a T, and so on and so forth. It will be going in this direction from 5 prime to 3 prime. And this is called the leading strand. That is the leading strand. That is the leading strand. That is the leading strand. But in the bottom, you know it's anti-parallel. It's two snakes, one head up, one tail bottom. If replication is happening, remember what the law says? The law says you can only replicate 5 prime to 3 prime. From 5 prime to 3 prime. But what about here? What's going to be the direction of my replication? It's going to be this way. Excellent. So that's going to be my direction of replication from 5 prime to 3 prime. The strand that you have your normal direction, 5 prime to 3 prime, that's your leading strand. The one where you have the reverse, that's going to be the lagging strand. The lagging strand. The lagging strand. And this lagging strand is very important because it's going to replicate for me, but in cut up segments, in fragments. In cut up fragments, known as Okazaki. Okazaki fragments, named after the Japanese scientists who discovered them. These are known as Okazaki fragments. So it will be going 5 prime to 3 prime, stop. 5 prime to 3 prime, stop. So these are Okazaki fragments. Okazaki fragments. Then a glue DNA, known as DNA ligase. DNA ligase. The DNA ligase is the glue that will connect together all the gaps between my fragments. So it is the lagging strand that will have my Okazaki fragments, these cut up, chopped up fragments, and they are connected with DNA ligase. They are connected with DNA ligase. And transcription, and this process is called discontinuous. So replication is discontinuous. Replication is discontinuous. Questions? Well, you must understand that replication will never happen except 5' to 3'. You must always remember that you have a leading strand where everything is happening normally. And you must also understand you will have a lagging strand where everything is cut up, and we're going to stick and glue everything using a DNA ligase. Okay, from now on, I will publish the study guide the weekend before the exam. So now we can learn. Yeah, so we can learn because no people like, I'm talking to myself.
Your study guide is too long for that.
Yeah, okay, but that's the way it's gonna be.
All right, I have a question.
Yes.
The pink line that's connecting, you said is what?
That is the DNA ligase. The DNA ligase is glue, ligating and connecting together all the, you see this purple, these fragments have to be connected. But after they're connected, they are back green again. So they go connected like that. They're all green. They're all green. They're all green and they're all connected. Then you have a double. You see it like that. They're gonna all be doubled up, and then you're gonna divide, and you're gonna have your two cells terminally.
So this has to happen in order for mitosis to happen.
Definitely.
Okay.
If this does not happen,
And
there's
what's it called?
apoptosis.
What is this called? What does it start with? H? What's the word of this? Helicase.
Helicase is the enzyme who unwinds
Yeah.
and allows us to separate the strand and start this transcription, this replication. So that is replication. We are done from here. Here the circle is like, the arrow is like that. So we make from DNA, DNA. We make from DNA, DNA. From DNA we double up DNA. We double up DNA. We double up DNA. So the more DNA we make, we are getting ready to divide. You cannot divide a cell that does not have enough DNA for the next round. Yeah, excellent. Okay, let's now go to the next step. Now we left here, and remember the DNA replication is happening inside the nucleus. Now we're gonna go from the nucleus to the transcription. So now let's see how transcription happens. Transcription is very similar to the replication, but we depend on the DNA, but we do not use a DNA polymerase. We use an RNA polymerase. So we're not going to be adding a T, we're going to be adding uracil. We're going to be adding uracil. So let's say this is my DNA. I need a particular protein. I need myoglobin. I need myoglobin. I need, let's say, hemoglobin. Hemoglobin. I need, for example, catalase. And that is very important to reduce oxidative stress. So my DNA has many regions. This is the region for myoglobin. This is the region for hemoglobin. And this is the region for catalase. This is the region for catalase. So now if I want to now transcribe, if I now want to if I want to get, this is for, purple is for myoglobin. Blue is for hemoglobin. And orange is for catalase. If I want hemoglobin, I'm going to unwind this region. Using helicase. So helicase is going to unwind this region, expose for me the myoglobin part, and everything else is going to stay attached. Because I need myoglobin. I need myoglobin. This is hemoglobin. And this is catalase. So now what will happen is, that's DNA obviously, that's DNA. That is DNA. The bottom strand is DNA. And the top strand is, the bottom strand is DNA, the top strand is?
DNA?
Why do you think it's RNA? Helicase is the enzyme who unwinds the zipper. It unwinds it for us. That's all it does. It separates them from each other. But this strand, what is it? If the bottom is DNA, how could the DNA be binded with RNA? Remember the DNA is double helix? Is double? It's DNA. So DNA in the bottom and DNA at the top. So now I open DNA in the bottom, DNA in the bottom, and DNA at the top. Then my polymerase is gonna come. My polymerase is gonna come and it's gonna read the whole myoglobin part. So this green part, translating for me, for every U, you get, for every A, you get a U. For every G, you get a C. For every C, you get a G, and so on. And then we're gonna use a DNA-dependent RNA polymerase to get the green. And then when that green, this is all happening inside the nucleus. Here, that's transcription. And then this green part comes out. This green part is known as the premature, is known as the premature nascent mRNA. Premature nascent mRNA. Premature nascent mRNA. I cannot leave it like that. So now my mRNA looks like this. That's my mRNA, that's part A, that's part B, that's part C, that's part D. But the problem is between them are the introns, are these regions that I do not want, are these regions I do not want, these regions I do not want, these regions I do not want. So what I will do now, after I transcribed my mRNA, I'm going to do post-transcriptional modification. What I'm going to do is, what I need, the purple I need is known as the exon. That's the exon A. That's the exon B. That's the exon C. That's an exon D. So all I need is the exons. The ones that I don't need in the middle, these are my intron. Introns are bad. These are my intron. These are my introns. So what I'm going to do, I'm going to cut them out. I'm going to separate them. I'm going to separate them where I get all my introns on their own. That's the first intron. That's the second intron. That's the third intron. And I'm going to have all my exons together. Exon A, exon B, exon C, and exon D. So now it's ready. After I excised and removed all the introns to remove them out, after I excise them, this is known as post. What is this known as? Post transcriptional, excellent. Post transcriptional modification. So I modified, I removed all the introns. So many students, they think the introns, because you say introns, they get to stay in the final mRNA transcript. No, the ones who stay in the final mRNA transcript, the final mRNA transcript, these are the ones who stay in the final mRNA transcript, these are your exons. Excellent. But the ones who get left behind, because these are going out now, these are going out to the ribosome. Ribosome, we said, is in the cytosol. Is in the cytosol. The ribosome for translation. So the exon is exiting the nucleus, is getting out. This is all happening. The mRNA transcription is happening inside the nucleus. This is 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 the nucleus. And you have these are the nuclear envelope, and you have the DNA. You have the DNA, and you do the post-transcriptional modifications here, and only the exons exit. That's why we call them exit, because they exit the nucleus and they go to the ribosome for translation. They are VIP. They are going on a cruise. They are going outside. They are exiting. But the ones who stay behind are the introns, because they are useless. So now what does the cell do to that? The cell will do something known as alternative splicing. Alternative splicing. So now I'm going to add stuff from the lecture that the people are not paying attention to on the exam. So it's not just the study guide. You have to pay attention, and you have to go home and rewatch this lecture. I'm going to post it to you and rewatch it, and I'm going to add questions from here. So alternative splicing is when all the cut up introns, we recycle them. We add them together like for alternative splicing. It's called alternative splicing is when we add up all the chopped up introns. All the chopped up introns are going to be added together in order to give me a new form of mRNA that I can convert into exon, and now it can get translated to give me some form of useful protein. So introns, they get cut out, left behind temporarily. Then the cell will do alternative splicing to reassemble them together. And recycle them, make some useful mRNA transcript, and make some useful DNA off of that. So that's transcription. After I transcribe, then I translate. How do I translate? I take my exon here. I go into the ribosome. So now that's let's say this is my final mRNA transcript. That's my final mRNA transcript. And it's made up of 3 codons. Always the first start codon, known as the start codon, is always known as AUG. And it also codes for an amino acid known as methionine. An amino acid known as methionine. AUG is a 2-in-1. AUG is a 2-in-1, is a start codon. AUG is a start codon, and also codes for methionine. So what happens is this final mRNA transcript, the ribosome is free floating in the cytoplasm. So the ribosome, it has two subunits. The smaller subunit in eukaryotes is known as the 40S subunit. That's the small subunit. That's the small subunit. That's 40S, or the small subunit. Then the large subunit is going to bind on the top. The large subunit is going to bind at the top. And the large subunit has 3 gates. It has 3 gates. It has an entry site known as the A site, a polypeptide site known as the P site, and an exit site known as the E site, EPA. So what happens is a 3-leaf clover like mRNA known as RNA known as the transfer RNA, tRNA, is like 3-leaf clover. It will come, it looks like this. Looks like that. This is AUG. It looks like this. It has an anticodon here. So with A is going to have U. With U is going to have A. With G is going to have C on its head. On the tail is going to have methionine, the amino acid, AA, the amino acid. So it's going to enter in the entry site. It's going to bind the start codon. Then it's going to shift to the polypeptide site. It's going to hold its methionine here. Then a new tRNA is going to enter with its amino acid, and then there will be a shift of amino acids from the P site to the A site. Then the transfer RNA that was in the P site is going to exit, and this one that was on the A site is going to shift to the P site, and now it has 2 amino acids. A third transfer RNA is going to enter, and it's going to be a shift from the P site to the A site, connecting the 2 previous amino acids to the third amino acid here, and another shift will happen. So now you will have a transfer mRNA with 3 amino acids, and the 3 amino acids are going to wait for the fourth tRNA, and the fourth tRNA we're going to transfer from the P site to the A site, or long string of beads, and we're going to keep doing that to accumulate a lot of amino acids in our primary sequence until we reach a stop codon. The stop codon is you go away, you are away, and you are gone. Once the ribosome reads one of these 3 termination sequences, they code for no amino acid. They code for no amino acid. They just terminate the sequence, and then you have a polypeptide sequence. That polypeptide sequence is like a string of beads connected by peptide bonds. And that is known as the primary structure, first degree. To go from primary to secondary structure, you need hydrogen bonding. So you need hydrogen bonding to go from the primary structure, into the secondary structure. And to go from secondary structure, you're going to need even more extensive hydrogen bonding, and you will get a tertiary structure like myoglobin. Myoglobin is an example of a protein in a tertiary structure. And to go from tertiary structure to quaternary to quaternary structure, you will need ionic bonding. So that shows you you're going to need different types of bonding to fold upon yourself. Ionic bonding. In order to fold from primary structure to secondary structure to tertiary structure to quaternary structure, you're going to need more extensive bonding so you can fold upon yourself. Why did Christian Anfinsen, the scientist, gain Nobel Prize? He got Nobel Prize because he discovered that what is the most important form of our protein? Do you think it's the primary structure or the secondary structure or the tertiary structure or the quaternary structure?
Quaternary?
Quaternary. That's what everybody used to think, that it is the quaternary structure. But he proved that actually it's the primary structure. Everybody knew that to go from primary structure to secondary, you're going to need extensive hydrogen bonding, and you're going to get alpha helix and beta pleated sheets. Then to go from secondary to tertiary, you're going to need more hydrogen bonding, and you're going to need hydrophobic effects. And to go from tertiary to quaternary, you're going to need more hydrogen bonding and more hydrophobic effects plus ionic bonding. He said if you mess up with the pH, and you mess up with the temperature, if you change the pH and if you change the temperature, you're going to cause the unfolding. So we're going to unfold from a quaternary structure to a tertiary structure to a secondary structure to a primary structure. But he said if you restore the optimal conditions again, so now you're no longer messing with the pH and you have your regular optimal pH, you have your regular optimal temperature, it's going to refold back up to 4 again. So he proved that it is the primary structure, because after we returned to the primary, we had no quaternary anymore. But after we reset the conditions, so now the pH is not too much, is not too low, the temperature is not too high, is not too low, it's optimal, then it refolded right back to the quaternary structure. So he proved that it is the primary structure that's the manuscript. That is the manuscript for your quaternary structure and your tertiary structure. Yes?
But is that for some, not all proteins going through denaturation, whatever you call it?
What do you mean? What do you mean?
So like if you heat up an egg and the egg white turns boiled, you can't
Yeah, because in that case, the heat was so much. See, remember when we talked about the types of bonds, ionic bonds, we talked about the hydrogen bonds. These are all hydrophobic effects. These are types of bonds, but they're weak bonds. It is the covalent bond, like the phosphodiester bond, like the covalent nonpolar bond, the covalent polar bond, the glycosidic bond. These are the strongest types of bonds. But when you heat up a boiled egg, it is so hot that you broke the hydrogen bond, you broke the hydrophobic effect bond, you broke the ionic bond, and now you even broke the covalent bond. That's why you cannot replace. So he's talking about the temperature and the pH that were enough to break the weak bonds. So it is the weak bonds who will make the quaternary structure, the ionic bond. You don't need a covalent bond to go from primary to quaternary. You don't need covalent bond. It is just those weak bonds. You see hydrogen bonding? I put hydrogen bonding and hydrophobic effect, and hydrogen bonding, hydrophobic effect, and ionic bonding. These are all weak bonds. So when you heat it up, they're all gonna break and we're gonna denature back to primary. But if you go to the level of egg yolk, heating it up, that is so hot that even the peptide bonds are broken. The phosphodiester bonds are broken. The glycosidic bonds are broken. So now you're destroying even the constituents. But he's not talking about that. He's talking about just in this structure. Okay? So just he's proved that these weak bonds are what give the protein its conformational structure of being quaternary. And you can reverse them back to primary. But if you reset everything back, it can refold right back up to the quaternary degree again. So that shows you that the primary structure is the manuscript, for the protein. And then when the protein is made, the same way we had post-transcriptional modification, we're gonna have post-translational modification. Okay? Post-translational modification. Yeah, we're gonna take a break at 4. So it's 4? Okay. Alright. We're not gonna come back to this room because the slides are not working. The slides